Hello everyone. I'm happy to welcome you to this webinar, Introduction to dMIQE Guidelines for Reproducible Digital PCR Experiment. My name is Alexandra Martin. I am Global Product Manager at Stila Technologies, and I will be your host for the next 30 minutes. Our speaker today is Vandesompele. Joe is a professor at Ghent University in Belgium and group leader at the Cancer Research Institute in Ghent, where he co-supervises the OncoRNA lab. Joe is a giant in digital PCR with multiple hats. He has led many initiatives and co-founded 3 companies. He has authored more than 250 publications and has an H index of 99 with 78,000 citations. We are very honored to have him today with us to speak about an important topic for the digital PCR community, which is how to design reproducible experiments. For that, the dMIQE guidelines were written, and Joe is one of the lead co-authors, so there is no better person to explain what the dMIQEs are and how they can be applied to your own research. This webinar will be in listening mode only. If you have questions during Joe's presentation, please type them in the chat box at the bottom right of the window. We will save 5 minutes at the end of the presentation to answer the questions. That said, I will now leave the stage to Joe.
Thanks very much. Hello, everyone. I'm happy to be invited on digital dMIQE guidelines. All right. So the title you know by now. Maybe one quick introduction slide on myself, if I can advance. Yes. So indeed, I'm an academic professor, but I'm also the co-founder of several companies, and one is particularly relevant for today. It's called Pixlens, where we develop PCR technologies, including universal probes for digital PCR. And we'll come back to that later in my presentation. So, all right. Let me share a frustration I have, and maybe you also feel sometimes frustrated with the scientific literature. Because if I read a scientific paper, often I have the feeling that important information is missing, or that people, the authors, do not adopt best practices. And that's really frustrating because we all want to advance science. And what is the result of this? Well, this is one example, a respected journal, Science Translational Medicine, in which the editor, him or herself, expresses a concern about a study that was published in his or her own journal. And I cite here that the procedure for performing digital PCR was incorrect in the supplementary materials and methods. So that's really kind of a shame that the editor of his or own paper has to warn an expression of concern. But even more problematic issues can happen in that the paper scientifically must be retracted. Several examples, I just showed 2 here. And one paper in which the editorial board expresses 5 concerns before they retracted the paper, that there were discrepancies in the description of the research reported, discrepancies between the availability of the data and the research described, but most appalling, incoherent, meaningless, and or irrelevant content included in the article on applying digital PCR to supposedly advance science. Another example is not a published study, but a preprint in which the authors used a human DNA assay to do quality control for RNA in which they quantify viral targets. Here you see the comment of one of the community members on PubPeer that commented on this particular flaw in that study. And finally, an example, I often act as a peer reviewer of manuscripts. This is an example of digital PCR data I had to review. Clearly, the threshold setting is incorrect, where you see that the threshold, these partitions are called positive, and they're clearly not. And the performance of the assay is particularly poor. You see a lot of rain, so definitely not adopting best practices to set thresholds or to optimize the assay. Just to give you a flavor of all the issues you can encounter in the scientific literature. So this frustration I share with many different co-authors, and 15 years ago, we developed the dMIQE guidelines, minimal information for quantitative PCR experiments, in how to design, execute, report, and analyze quantitative PCR data. Here you see the front page of that particular study. It was published in Clinical Chemistry 2009 and heavily cited. But despite a lot of citations, there is still a lingering problem. So before I continue on the problem, I just want to indicate what that historic MIQE guideline or guidelines particularly are. It's actually a community-driven set of guidelines that describes essential and desired information in literature. It's about 9 chapters depicted here on the left, and the checklist is depicted on the right, and it really serves 3 purposes. It's for authors, it's recommendations, guidelines, how to design, execute, and report on quantitative PCR. It's for the reviewers to have a better and transparent understanding of what the authors did, how they came to conclusions, a kind of a quality control check. And finally, also for the readers of the scientific study, if it's eventually published, to have a very transparent understanding of what was done, what controls were used, how the authors came to the conclusions, and if needed, how the readers can reproduce the study. It's a lot of effort to come to 85 parameters, and that's often the critic we receive, that it's really a lot of work. And to some extent, this is true, but if you know how to do your methods, I'm pretty sure that at least half, if not more, of these parameters, you can easily say, yes, check, 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 I did that, or I didn't do it, and because of this reason. So if you really know how to master quantitative PCR, I will come to digital PCR in a minute, it's not too much work. It seems daunting, but it's okay. One of the recommendations is to share data. Historically, we recommended the RDML, or the real-time PCR data markup language file format, but that was never very popular because it's XML-based. Authors, users were not very comfortable. So very recently, actually last year, we came up with a very simple format under the leadership of George Kalin and mainly spearheaded by Andreas Untergas, a very simple spreadsheet format to share quantitative PCR data. But hey, this is about digital PCR. So good news, a digital PCR data format is coming soon, at least as a scientific paper. For those of you that already use the Stellar Technologies NIO Plus or QIAcuity instrument, you can already export and share your data under the DDES format, standing for Digital PCR Data Essential Spreadsheet format. And really the purpose is that in the near future, we all share digital PCR data under this format. So everyone can reanalyze and hopefully come to the same conclusions. Not only reviewers, editors, but especially the consumers of the scientific literature, your readers. It's also for a plea for the digital PCR instrument manufacturers and software to make their software compatible so the data can be easily shared and within the community. So that brings me to an important question. Is there a reproducibility crisis? And not the only one. 8 years ago, asking or surveying 1,500 researchers worldwide, and it's appalling to see that at least half says, yes, there is a significant crisis, and another 40% says, there is a crisis, but it's not that big, there is a slight crisis. So summarizing, there is a problem. So what is the fundament of this problem? What factors contribute to the reproducibility crisis? Selective reporting, number one, methods or code unavailable, raw data not available, and insufficient peer review. And guidelines really can help address all these concerns, especially for the reviewers. If you're not an expert in digital PCR, but you can see that they adhere to a minimal information checklist, as a reviewer, as a non-expert, you can at least have a guidance in how to evaluate the quality of a digital PCR study. So minimal information guidelines is not a new phenomenon. This is a paper from 1985 in which minimal information guidelines were published for animals and husbandry methods. Quite cool, it's the first minimal information list. Since then, 197 studies came up with minimal information lists. And you see them progressing in time, and digital PCR actually has 2. In 2013, the original digital MIQE guidelines, and then in 2020, an update of digital MIQE guidelines. So really, it is important, and authors are starting, and scientists, to realize that it's important to come up with guidelines for more informative design, execution, and reporting of, amongst others, digital PCR experiments. So a little history about the dMIQE and the dMIQE guidelines. As already shown, the original frustration culminated in that paper in Clinical Chemistry in 2009, along with the data format, RDML, which has been abandoned now and has been superseded by RDES. There was a small update in 2011 on some frustrations that the dMIQE guidelines insisted on disclosing primer and probe sequences, but commercial companies were somewhat reluctant to do so because it's their bread and butter. So we came to a compromise that the context sequence of the amplicon was good enough to reproduce and do quality control of the data. And then finally, the dMIQE guidelines in 2013 with an update in 2020, and the data formats, very simple spreadsheet data formats last year and coming this year for digital PCR. So why did we come up with an update in 2020? Is it really old wine in a new bottle? I would say no, because 7 years between 13 and 20 is really a long period for a young and evolving technology like digital PCR. In the beginning, there were single color, 2-3 color instruments, and now we have 4, 5, 6, and Stella Eve has a 7-color instrument. There were studies and instruments with 1,000 partitions. Now we have easily 20, up to 100,000 partitions, and even more. And importantly, more new powerful applications came along. Drop-off assays, linkage or DNA integrity assays, a higher order multiplexing, where you quantify more targets than fluorescent detection channels in your instrument. So to accommodate these new evolving applications and instrumentation, we had to come up with new guidelines. Importantly, it's a much simplified checklist. It's only 52 parameters. I will show that in a minute. New factors that we now learned are important, and we ignored elements that we say, maybe that was not that important in 2013. And also the support for the new instrument manufacturers is integrated in the digital dMIQE guidelines update 2020. Importantly, we came with unified nomenclature. I just show you a few examples here. All the details are in the paper. Importantly, we talk about the partition, not about the droplet or the chamber, the separability between the negatives and the positives. That's called peak resolution. And we no longer speak about one-dimensional 1D and two-dimensional 2D plots. No, we say one color and two color plots or multicolor plots, just to give you a few examples to make sure that we all speak the same language. Peak resolution, quite simple. It's all about the width of the distribution of the negatives and the positives and the intensity of the positives and the negatives, and with a simple formula, and there are many out there. This is one from Anton Lievens, published in PLOS ONE in 2016 that we use, but many software developers actually have integrated some kind of peak resolution in their software. And I think it's important because it says something about the quality of the assay and how sure you are to discriminate a positive from a negative partition. And that's incredibly important for digital PCR because it's all about the quantitative accuracy in discriminating positives from negatives. Already talked about higher order multiplexing, where you can quantify more targets than you have fluorescent detection channels. Different strategies are possible to do so. You have amplitude-based multiplexing, but more recently you have ratio-based multiplexing depicted on the left, which is beautifully summarized in Alexandra Wales' review paper in BDQ in 2016. And you have color combinations, which were originally published or shown to work for quantitative PCR, but now a very beautiful paper from the Stella team, which I think is still pending review, but the preprint is available, how to do so, and how we can really leverage the multiplexing capabilities to a higher level. Of course, higher order multiplexing really requires a lot of probes, and some people may say, well, that becomes horribly expensive. Not necessarily. There are solutions out there. Just a shameless plug for one of the solutions that we have developed called rainbow probes. If you want to know more about it, please scan the QR code or go to our website at Pixlens. It's 6 probes that can quantify any target and really can make multiplexing and higher order multiplexing very cost-effective for you in your research. So just as an example that these universal rainbow probes, they work out of the box. This is some data on the NIO Plus instrument that my colleague generated. On the left, you see a mutation assay where we use an allele-specific primer modified so the universal probe can detect the amplicon with exquisite specificity. This is a BRAF assay. And on the right, you see the same wild-type BRAF assay in two different detection channels simply by substituting one of the primers, because it's the primer that is recognized by the universal probes. So higher order multiplexing really becomes within reach for the scientific community. So coming back to the dMIQE checklist, these are again 9 chapters depicted here, from where does your specimen come from? How was it processed and treated? How did you extract the nucleic acids? If you do gene expression, how did you do reverse transcription? How were the controls made? Your oligonucleotides, how did you design and validate them, etc., etc. And it comes with a very simple Excel-like spreadsheet. And you only have to indicate in one column whether this particular parameter is important or not, yes, no, and if needed, you can add in the third column some complementary information for the readers or the reviewers of your scientific publication. So it really aims to be a supplementary table to a manuscript. And if you've done it carefully, really it's not too much effort to do so, but you really are very transparent in how and why you did your digital PCR experiments. So apart from going into the details of the dMIQE checklist, I want to indicate that the paper really comes with a richness of information, also considerations to design and execute your digital PCR experiments. Much to information within the short frame of a webinar, but I want to indicate that with respect to assay design and optimization, there's a lot of information, how you can exploit a 1-color and a 2-color plot to understand that the information is correct, how to optimize primer and probe concentrations, how to optimize the annealing temperature using so-called gradients, or how important it is to use a sample that is as close as the final clinical specimens or samples, if it's, for instance, a soil or a plant that you want to study. So don't underestimate the matrix effect of the real sample compared to synthetic or contrived samples. A lot of information is actually embedded in that dMIQE paper. But also when it comes to multiplexing, how you can discriminate artifacts like bleed through, in which one fluorescent signal is also apparent at a lower level in the other fluorescent detection channel, versus reduced specificity, whether you have probes, for instance, wild type versus mutant, that may not be 100% specific. We come with recommendations how you just can discriminate between these two. And if you do digital PCR and you inspect 2 color plots, how you can be sure that the partitions are at the expected locations. Just to give you one example, and there are many other strategies available, but on the left you see a non-competing assay where you really need to have all your 4 partition clouds on an orthogonal squared level. So the double positives in orange here need to have the same fluorescent intensity as the single positive blue and green ones. And the blue and the green ones need to be at the same level as the negatives. So indicative of no bleed through or no cross hybridization. Very different if you work with a competing assay. 1 set of primers with 2 probes that, for instance, detect the wild type and the mutant, where you have the double positive in between, like an arc in between the single positives. If you see the middle plot and you work with a non-competing assay, so 2 different primer pairs, you know something is wrong, or vice versa. And finally, a drop-off assay where you know where you can expect the double positives or the single positives or the in-between partitions. So knowing upfront where to expect and then verifying that they are at the right location is a very important step to trust your data. Temperature gradients, many others have shown this before. On the left, an example where the temperature really has a large impact on the final fluorescence intensity. On the right, an example where this is not the case, where you see that this assay is very robust against temperature fluctuations or different temperatures. So it's a very neat tool to optimize and understand the optimal temperature for your assay. We typically take the highest temperature with reasonably high peak fluorescence, peak separation. You can really use that to optimize your assay. This is just one example here on the left, a non-competing assay. I already explained that the double positive orange are not at the right location, so they are lower in the blue channel. By optimizing under different ways, it's about primer concentration, it's primer TM, sometimes changing the master mix, you can really get that double positives at the location where you expect them, which you see here on the right side. Just to give you one example that you can play with primer concentrations, this is an assay in which we expected copy number ratio of 1. It's clearly below 1, so there was a dominant and a permissive or slave assay, I would say. Well, here we reduced the primer concentration of the dominant assay from 900 down to 100 nanomolar, and you see that we approach the expected ratio of 1 by playing with primer concentrations. And this is the same experiment now showing copy numbers where we have suboptimal concentration of the copies with the dominant assay present. But if we titrate that dominant primer, go to more favorable conditions for the multiplexing to work, we approach the expected copy numbers. So it is important to do this kind of validation and verification experiments. The dMIQE guidelines end by a lot of considerations for design and execution of the experiments, including data analysis, how to do manual or automated clustering, and there are some caveats in there, but also how to do advanced data analysis, for instance, to use multiple reference genes to do a proper propagation of replicates, so propagation of the uncertainty, but also how to do multiplex data analysis independent of the particular experiment design. At Ghent University in the Digital PCR Center, we have a lot of software tools available. For those of you that are interested, please have a look at our website, dpcr.ugent.be. We have a lot of tools free for you to use to improve your QC or your more advanced data analysis. And finally, the good news, I'm almost there. I think it's not a secret, it's not a surprise that digital PCR is really taking off tremendously. The increase in number of publications is year on year increasing. We have last year 650 publications, that's almost 2 per day, applying digital PCR. But the good news is also about a quarter of these studies, they do cite the dMIQE guidelines. And other studies have shown that if you cite minimal information guidelines, that you're much more adherent, compliant, and transparent in how and why you did your experiments. So we're now approaching about to an adoption rate of 1 in 4. I think it can definitely improve, but it's definitely very encouraging. So how can it improve? How can we increase the adoption rate? Well, actually, as a scientist, lead by example, by just doing it, showing the example to the readers of your papers or to your colleagues. Endorsement by scientific journals. On the right, I give you a few examples of journals that endorse the dMIQE guidelines, sometimes already also the digital dMIQE guidelines, not yet. So there is work of the community to enforce or to ask journals to do so. Please spread the word on your poster and scientific conference, in workshops, education, convince about the necessity, like showing what errors you can make, or even that it can lead to an expression of concern or attraction of your paper. And finally, last but not least, is the role of the instrument supplier. I'm very grateful for this event of today by Stellar Technologies. It's really, they embrace the concept and they are educating their users. So to come to the conclusion of my webinar, I think to fight the reproducibility crisis, we need to transparently report all important information. Minimal information guidelines like the dMIQE can help achieve that goal. Checklists are actually quite easy to complete, and you simply add them as a supplemental information to your paper. And it's not about the others, it's about us. We all carry responsibilities. Brings me to my final slide. I want to acknowledge the dMIQE and the dMIQE working groups, and especially Stephen Bustin and Jim Huggett, who are really the leaders in the dMIQE and the dMIQE field, all endorsers of transparent and reproducible science, and again, Stellar Technologies for pushing the boundaries of digital PCR and endorsing so heavily the digital dMIQE guidelines. And with that, I'm happy to take any questions.
Thank you very much, Joe, for the very interesting presentation that has triggered already many questions. So we will save the last 5 minutes to answer as many as we can. And if we don't have the time to answer to your question, then we will reach out by email after the webinar. So I'm gonna go through them. So first in, first out, right? So I will start by the first we received. It's about the peak resolution. Although the peak resolution is integrated as an estimated number in some platforms, the calculation remains difficult from data obtained with other instruments. Is there any tool available for the calculation of the peak resolution, and which sort of raw data you need for such calculation?
Yeah, good question indeed. It's a concern that companies have different metrics. So typically from the instrument software, you can only use the compare within if you use the same software, but you can export raw data. I think all platforms nowadays allow that. So the type of information they export is good enough. And the paper I cited from Anton Lievens in 2016 actually has an R script. There will be a Shiny app on the digital PCR consortium website as well. But if you're a little bit R savvy, you can use that script. And if you can export in DDES file format, we will allow direct import to do peak resolution. But it would be good, it's a very fair comment, that the different software and digital PCR instruments providers would come to a unified definition of peak resolution. A valid concern.
Okay. Next question. Do you have recommendations for acceptable statistical similarity between replicate runs using the same samples? I've been looking at different coefficient of variance, but it is hard to know what is too much variance between runs.
Yeah, again, very good question. It really depends. And I'm sorry, I cannot be more specific, but there is a concentration dependency on variance. We all know that if you have a high concentration, that typically the reproducibility is better. This is true in any quantification method, including digital PCR. So there is no gold cutoff, but what is perhaps important, if this is really important to you, what has been agreed upon many different organizations and bodies is actually the limit of quantification typically limits reproducibility at 35%. This is not cut in stone. The 35%, 40% is typically accepted as below that. We say it's less reproducible. Above that, it's very reproducible. But we all know with digital PCR that we can easily surpass that. So if you have a good assay, if you have sufficient concentration of your nucleic acid in the digital PCR, you really come to 10, 5, even 2%, depending on the lambda, so the partition occupancy of your experiment. That's really a very important driver of CV. But as a general guideline, I would say try to work above limit of quantification. If not, you're in the range of detection where you can say yes and no. 35% seems an acceptable CV, but know that you can do much better with digital PCR.
Okay. The next question is about the number of partitions. Do you have a recommendation about the minimum number of partitions for having good digital PCR data?
Yes and no. I think 10 to 20,000 is a good ballpark number. You see, most platforms can easily do that, even if you lose some partitions. But again, it really depends on your application. For some applications, you can get the same quality of data with far less partitions. For other applications, you need more partitions. So sometimes for some applications, 100,000 may be advantageous, and you can do that either with chips or formats that can immediately do that, or you can bundle, do some kind of meta-analysis of different replicates. But on average, most of the studies, most of the applications, most of the questions can be addressed between 10 to 30,000 partitions rather easily.
Okay. There is another one that is maybe more for Stila. For higher order multiplexing, is there a universal probe available for the 7th color of the Stila instrument? So I can comment on this one. Yes, Stila does support higher order multiplexing for all 7 channels of the NIO Plus through the use of the new Flex probe, which are also a set of universal reporter optimized for the use in RubyChip. And with this probe, we also offer surface assay, and they are being conceived keeping the dMIQE guidelines in mind, of course.
Excellent. Good news.
I just maybe take one last question. Okay, so this one probably for you also on the multiplexing. Could you explain how the probes for multiplexing are designed?
Would that be target specific or universal? For target specific, I think there is no secret. I think the primers are the more important elements in a multiplexing, that there is no off-target or cross-homology or non-specific amplification occurring. That's relatively easy to check. You can do a singleplex, you can do the multiplex, and the quantitative results should be the same. And the probes are merely a detector. So I think probes have not a real impact in multiplexing, except for the fact that if you start combining too many classic hydrolysis style or hairpin style probes, that your background may become too high. And that's where universal probes come in. You keep the number of probes low to 4, 5, 7, depending on the application and the instrument. You keep the background low, and then you can do multiplexing higher order multiplexing. But the sequences for the universal probes are, of course, proprietary to the companies that supply them. I hope that answers your question.
I don't see any more comment on the questions. Unfortunately, we'll have to close the session for today. We have many questions unanswered and also still popping up. So that's good. That triggered a lot of interest, I guess. I will thank you again, Joe, for the nice presentation, and we will reach out to every person who didn't have the question answered. Thank you very much, and have a nice rest of the day.
All right.